but if we don't know something, we go to dictionaries. So the dictionary of the English language refers the mind to this origin, to old German or English Gemünd, meaning to remember in a way. So the very word mind comes from the word which is somehow related to the memory. And this was our assumption from the very beginning, that memory can provide a window to understand mind. I'm not saying that memory equals mind, but it's an important aspect and attribute of mind. The very important concept when talking about memory is the concept of plasticity, synaptic plasticity. This actually has been introduced, the term, by Leszek from the Nencki Institute, slightly recently recalled by Eric Andeli and Indodai and Mark Mayford. But originally, the idea that memory could be encoded in the structure of the brain and connections between neurons comes from Ramon y Cajal. This is actually interesting that nowadays the term synaptic plasticity is kind of almost equal to electrophysiological properties of the synaptic transmission, whereas origins are in the morphology, and I will be referring to this morphology quite often. How may it look? My very primitive visualization of this concept is, this is the network of neurons in the brain, a very small piece of it. You know, there are 85 or 86 billions of neurons in the human brain and thousands of connections between them. And the engram, the memory trace, what we remember, is then the idea that it's a path of a signal, of the information that travels between neurons. A neuron excites another neuron, and excites another neuron, and so on. And then the distinction, so it's a network, and the distinction is at the level of the synaptic connections. When we talk about excitatory synaptic connections, we talked about glutamate as the major excitatory neurotransmitter. The vast majority of neuronal excitation is mediated by glutamate, released from the presynaptic site, traveling from the synaptic cleft, and then binding to the receptors located in the postsynaptic domains. And those excitatory synapses are located on so-called dendritic spines. There are small protrusions, majority, I know 88% of excitatory synapses are located on dendritic spines. That's good, because this is, allows us to investigate this morphological structure. We will come back to that. Our contribution to the field was to introduce molecular biology to this picture in the late 80s, and especially discovery of gene expression in learning. The concept was that during changes of this connectivity, starting from Cajal, Fukonorski, and many other giants of the field, the hypothesis was that there will be also changes at the level of cell nuclei. There will be changes in the gene expression evoked by behavioral training, by learning experience in animals. And if we identify the genes, we may approach the understanding what is going on here. Because in principle, the changing the efficacy of this connectivity may come from the increased glutamate release that could be regulated by various levels, better traveling of the glutamate through the cleft, different receptors or modified receptors, more receptors, if you talk about increased efficacy of the synapse, more excitability of the postsynaptic neurons, many different things. So the idea was that if there will be changes in gene expression, they will reflect what's going on here, somehow related to this. So we looked for the genes that changed upon behavioral training, and first of such genes we identified was CFOS. And CFOS codes for a protein, CFOS protein. There's a component of so-called AP1, activator protein 1, transcription factor. Binds to joint proteins, forms a dimer, and dimer binds to specific gene sequences. and then regulates gene expression. So one of the things we followed was to identify the genes that are regulated, inactivated neurons by CFOS. Interestingly, through over 15 years of work, we ended up with two major genes we identified to be activated. I'm sure there will be some other genes and many other genes activated by CFOS in activated neurons, but somehow we focus on those two. And interestingly, those two are actually the proteins encoded by those genes are functionally linked. MMP9 is matrix metalloproteinase 9. It's a protein produced inside the cells, packed into some vesicle, released outside. I will show you that also at the synapses, in so-called latent Prof form, in which a propeptide, the whole molecule is about 100 kilodalton, propeptide is about 10 kilodalton, that covers the enzymatic active site. It's a protease. It requires zinc in the active site, so it's called metalloprotease. And it's called matrix because when activated, it cleaves through some proteins that are in the extracellular matrix. This activation step, removal of a propeptide, unleashing of the enzymatic activity, is mediated by some other proteases that are probably not real. Then the activity is there, cuts through some proteins, either being in this space or even extending from the cells into the extracellular space. But then it's pretty fastly blocked, inhibited by protein like TIMP1, tissue inhibitor of matrix metalloproteinases. So in this way they link. In our hands, this takes minutes. After activation of neurons, after activation by glutamate, the excitatory synapses, it takes 5 to 7 minutes, maybe 10, to release the protein outside the cells. Then it's active for additional, I don't know, few minutes, 10 minutes, probably no more, because then the TIMP, which is also there, blocks. The protein that was shown there is beta-dystroglycan. This is the first identified target of MMP9 inactivated neurons and excitatory synapses, actually. And this is evidence for that. So we produce this MMP9 protein in two versions, active and non-active, in the recombinant baculovirus system. So they are pure proteins, as you see here. But these proteins were run also through a gel containing gelatin. And this MMP9, as well as another MMPs, there are over 20 MMPs, called MMP2, are gelatinases. So they have the ability to digest, to cleave gelatin, which is denatured collagen. So the gel is loaded with gelatin, the normal kind of lemony SDSP gel. And then the proteins are resolved according to the molecular weight. And then all this nasty stuff like beta-mercaptanol SDS are washed away. And the enzymatic activity recovers. And then the enzyme cleaves what is in its location. So it cleaves gelatin. Then the gel is stained for the proteins with Kumasi, so it's dark blue. And only where the activity digesting those proteins, the gelatin, is available, is translucent, is transparent. So we see as a white button. So we have an active enzyme, non-active enzyme, we can put on the culture. Here I put an active enzyme, and you see that this beta-dystroglycan, which is 43 kilodalton, reveals about 30 kilodalton digestion product. So it cuts here, and this product is visualized. We can stimulate, this is just the enzyme, we can stimulate by copulin that blocks inhibitory input to the neurons, reveals excitatory, secondarily to blocking of inhibitory because there's a disbalance in a way. Apparently is believed to be mostly synaptic. It also produces this cleavage, but in this case we treat this culture, it's a hippocampal neuronal culture, with adenovirus as a vector to deliver proteins, either control protein, beta-galactosidase, or TIMP1. So in the presence of the control proteins, there's a cleavage. In the presence of TIMP1, the cleavage disappears. So it's blocked. So it has to be MLP activity. And finally, we can also subject mice to episode of seizures with betadine and tetrazole that acts more or less as bicopulin, blocking GABA, inhibitory input. And after seizure, there's an increase of this cleavage. 5 minutes after the episode of seizures, and this does not happen in MMP9 knockout animals. So just missing a single protein, MMP9, abolishes the increase of this cleavage. I'm not saying that it's cleavage entirely, because this could be cleaved by other enzymes, but we believe that this increase of the cleavage within minutes after stimulation of excitatory synapses is driven by MMP9. What is beta-dystroglycan? It's very poorly studied in the central nervous system, but has been shown by Thomas Sidov that actually it binds to neurexins. And we show that beta-dystroglycan could be present at the excitatory synapses on the postsynaptic side. Neurexins are on the presynaptic side. The best known partners of neurexins are actually neuroligands, and neuroligin 1 at excitatory synapses. So they sort of span the synaptic cleft, connect both pre and postsynaptic parts through this protein bridge. Interestingly, Rui Peixoto and colleagues 2 years ago shown that MMP9 can also cleave neuroligin 1. Now coming to the memory. Memory could be studied in animals pretty easily, and this is an example of the system we are using to study memory. This is so-called IntelliCage, a system developed by Hans-Peter Lipp, our friend from the University of Zurich, already retired, but still collaborating with us. There's a group of mice staying in a large kind of a rat cage with a food delivered from the top and the liquid to drink from the corners. There are 2 bottles in each corner, this is important. And in order to penetrate, to get into the bottle, they have to make a nose poke, this small kind of gate. And then the system opens an access and they can leak. And we can follow what the animals, what the mice do in this system, because they are implanted with an electronic chip, so-called transponder, subcutaneously, and this chip, which is unique for every animal, interacts with a special antenna system in the corner that allows to score different activities. There's also air valve in this corner, which allows to emit an air pump. And this system can be used for very simple form of training and memory formation. In 2 bottles in 1 corner. So first we have in all 4 corners, in all bottles, plain tap water. And then we change the bottles in 1 corner to sweet water. And they start to prefer this corner. It's called place preference. If they go to this corner preferentially, it means that they remember the location of this sweet corner in the cage. And then we can put the air pump into this corner. And they start to avoid this corner. It's called place avoidance. So this is a very simple measure of learning that can be expressed as percentage of visits to specific corner. And we subjected the mice I mentioned, the MMP9 knockout mice, to this kind of training. And found an interesting difference that the MMP9 knockout mice had problems in acquisition of the sweet corner task. They were somehow delayed or deficient in learning the location of the sweet corner, though they didn't have any problems with distinguishing the air puff corner. Place preference belongs to the larger category of learning that could be called appetitive learning. Appetitive is expressed by approach to something. Food, I don't know, water, a partner, shelter. And then there's another group of learning tasks. They are called aversive, in which the animals, or we humans, try to withdraw, to escape, to avoid, or even freezing is a kind of form. If there is a mouse on a field and there's a predator bird, so the mice want not to be seen, so just freezes, doesn't move. This is also kind of avoiding the predator.